So this is the Space Maroon Empire. I am your Space Maroon host, and today we are talking about plants in space. Firstly, why are we talking about plants in space? Why does it matter? Why would we even try and grow plants in space? And there's multiple reasons, and those multiple reasons are, well, first we need food. Food for our astronauts who are on the ISS and who may or may not be going to the Moon, Mars, further. Currently, we've been sending food up and back and forth to the ISS using just shipments. We're flying it back and forth. That is okay for now, but we're going to need more food as missions become longer, and we're going to need to make longer trips as we make missions further away. So, especially to going to the Moon or Mars, these kind of tricks are prohibitive. So another benefit to growing plants in space is the concept of the advanced life support system, where we use not only physical chemical systems such as air filters and monitors like that. We also use biological systems such as plants to convert carbon dioxide to oxygen, to recycle our toxic waste and stuff. So this is important because as everyone should know at this point, we exhale carbon dioxide, plants use the carbon dioxide, respirate and release oxygen, and then we take in the oxygen. It works. It's been proven by Earth. And I think we can use that, and I think, along with NASA and the other space agencies, think that we can use that on the ISS to supplement what we already have in terms of life support. Finally, morale. Plants in space, even having a little plant on your desk, will make you feel better, and it'll help astronauts better perform their missions in space, their experiments and their studies. And that's great. I think we've thoroughly established why we're doing what we're doing. But now we have to talk about how, and that's going to be our little study question of the day. So as I'm talking about this, I'm going to list off a bunch of issues for y'all, and I'm going to try and address or give you the tools to address some of them. So think about how we would go about this, and if you see any issues that you pick up on, so any solutions that you think of, post them in the comments, and we're going to talk about it. Right. So among the issues that we were going to talk about, a lot of them are based on microgravity, which is the minimal, almost zero gravity that you experience on the ISS. These issues involve cell division issues, where when plant cells divide, you find a lot of chromosomal breaks or deletions and that kind of stuff that happen. Or we'll have gas exchange issues, where gases don't exchange properly because of gravity not acting the way it normally does on Earth. Signal transduction, where specifically calcium would flow from A to B based on gravitational pull. It just doesn't happen because of low gravity. You also get issues of nutrient delivery. How do we get food that is in water or in the soil into the roots, because they won't just flow in based like normal. And we also have the issue of ethylene buildup, which is just a byproduct of plants being grown in a closed surface, closed area, and air not cycling properly. Finally, light. We have to figure out a way to give plants the sufficient light they need, because they need to, they need light to grow for photosynthesis. So we need to figure out solutions to these problems. Now, one way to simulate gravity, which is one way to deal with the issue of minimal to no gravity, centrifuges. With a centrifuge, if you imagine how, as a kid, you would take a bucket, fill it with water, and you'd swing it around really fast, none of the water would fall out because gravity. We can use the same concept to simulate gravity, as if we had, to facilitate water flowing the way we'd expect it to. We can use centrifuges and in many experiments, including some done by the European Space Agency up in the ISS, have used centrifuges to simulate gravity, to see what the effect on gravity would be on certain factors. Like an example of an experiment like this is the Gennaro project that was run by the ESA. They were looking at the distribution of the plant hormones IAA and ABA, where to see how they would cycle, how they were distributed through the plant in different gravitational situations. So different amounts of G, if you will. So what they would do is put it in the centrifuge, along an axis, get the thing going. They'd let the plants grow from seed to full sapling, I guess. They would end the experiment, deep freeze it, bring it back to Earth to be studied for genetic expression and distribution of these hormones. Another experiment is called Gravity, where they were looking for the minimum forces required for the gravitropic movement of roots, so the movement of roots in response to gravity. So what they would do once again, put them along an axis, get the thing going, and then see which way the roots would curve. A lab out of Wisconsin, which worked with NASA, sent up a module to the ISS to see if they could grow Arabidopsis Thaliana, which is a thale cress, from seed to seed. So they would grow up to full plant adulthood and then see if they were able to germinate viable seeds. So the main feature of this is that the module was divided into two areas, the bottom one being the root tray. In this root tray were a series of porous tubes. These tubes, using negative pressure and capillary forces, if you remember from high school, the adhesive forces of molecules to each other and to wall surfaces would pull them along into wherever you wanted them to go, and then negative pressure being like a vacuum, so we're sucking them this way and they're pulling each other along, and it just kind of works going to where you want it to. Using a method like this to bring nutrients, specifically water, to, and then whatever the water's carrying, to the roots of a plant, and then that way you can force feed them, essentially. The last thing I want to address is phototropism, an alternative to simulated gravity, where we can stimulate plants to grow a certain direction using light. One study that the ESA did, which is the Tropy study, is examining a phototropic effect based on red light, where plants, a positive phototropic effect, where plant roots will grow towards red light. Now they had three different situations: control, which is no light, I guess, red light as a positive, then blue light as a negative. So the experiment was set up that they had the plants in a centrifuge would be spinning, and then they would have the lights perpendicular to gravity so that they could test whether the roots were growing in the direction of gravity that they were stimulated in, or in the direction of the light that they were being stimulated in. A lab out of Japan, from the Japanese Aeronautics Exploration Agency, or JAXA, proposed the idea of using insects as a supplementary food source. So they said that a diet consisting of soybean, rice, sweet potato, and komatsuna, which is a Japanese mustard spinach, would be acceptable, but would be lacking in animal protein and lipids. So to supplement this, insects should be used, specifically termites, drugstore beetle, hawk moth, and silkworm. Now, both silkworms and hawk moths have been used as food in Asia, specifically Japan, China, for many years. The Japanese authors of this study acknowledge that they are available in stores in Japan. The silkworm, we can use the silk that it produces for various things. We can make clothing, fiber repairs, all sorts of things. The hawk moth feeds on the leaves of the sweet potato plant, which is convenient because that's part of the diet, and it also converts most of the protein that it digests into animal protein, which we can digest, and then because it doesn't make a cocoon, more of it is converted into animal protein than the silkworm does, so it's more efficient for us. The silkworm is more beneficial in terms of the other products it produces, such as its silk, but the hawk moth is more beneficial in terms of diet. The drugstore beetle and the termite can convert cellulose into more digestible protein for us. So there's the benefit. They can eat plants and give us what we couldn't normally digest, the cellulose, as a digestible compound. This paper also puts forth the idea, which is a next step idea, of using the loach fish as part of our diet, which is a fish that's grown in rice paddies. This is an idea we would probably use in Mars exploration, but wouldn't be feasible on the ISS, where it's just not enough room. Finally, on the topic of recycling of materials, a paper also from JAXA has promoted the idea of using hyper-thermophilic bacteria. Hyper-thermophilic meaning that it functions in very high temperatures, 80 to 100 degrees Celsius. Why would we use them now? Because in a composting situation, like one we would have on Earth, you put compost in, it generates a lot of temperature, it's high heat. Now, in high heat, most bacteria tend to fail, but we can use these bacteria, and they will function they will activate in these temperatures, produce and work more efficiently. These bacteria are useful not only because they function at high temperatures, which is where compost really gets going, but also because they have other little tips and tricks that we can use. For example, these bacteria do not produce nitrate ions, they produce organic nitrogen compounds, which plants can absorb out of the compost. Also, they fix heavy metal ions, such as zinc, and that removes them from soil and they are no longer toxic to plants. Sodium concentration can become high because these bacteria do not lose function at high salinity. So either the compost has to be processed again to remove sodium, or we can use the compost for plants that require high sodium anyway, such as algae. And then the algae itself later can be used as compost. And I guess an extra thumbs up here is that these bacteria tend to trap odor compounds, which can either be toxic or just they stink. So go bacteria. So the authors believe that the bacteria will be safe for humans because once it's outside the 80 to 100 degrees Celsius range, they go inactive. So it's generally not an issue for us. Of course, it needs to be tested, but if it becomes an issue, they figure using a filter to remove the bacteria should be fine. Lastly, this will lead us to our question of the day. I asked you guys to keep an eye out for anything, any ideas you've had based on what I've said. So any issues that I brought up, any solutions that I had, any reasons why to grow plants. If you thought of anything else, you post it in the comments. We'll talk about it. My question for you though, can you think of any plants that would be suitable for growth in space? So consider nutritional benefit. So we need, if we're gonna eat these plants eventually. Consider benefits to study, so that they are easily examinable for scientific reasons. That we can look at this genome, we can look how it responds to this and that stimulus, and is it robust enough to grow in space? Because growing in the ISS is going to be a little bit of a challenge for most things. It's a challenge for us, but it'll be more of a challenge for plants. So we need a species that's robust enough for us to grow on the ISS. If you guys can think of one, you can post it below, and we can evaluate why it's a good idea, why it's not. Well, that's all that I've got for today. Thanks for watching. Make sure you guys comment, like, subscribe, the usual YouTube deal. And we'll see you next week. Space! This paper, there's just points where I just look awkward. We can just put pretty pictures over it. I'm just feeling super Bill Nye today. That's good. So, what? The hawk moth. Like, ah!